Good day, everyone! Today we are going to dive into the presentation of Experiment 5, the DNA extraction of plant samples. But first, let's discuss a topic that is kind of connected to it, which is the nucleic acids. Nucleic acids are a family of biopolymers essential to life as we know it. These remarkable molecules carry genetic information, making them universal across all living organisms, from the smallest viruses to the most complex human cells. So, what are nucleic acids? They are long chains of smaller units called nucleotides, and each nucleotide is composed of three key parts: a nitrogenous base, which can either be a purine or a pyrimidine, a 5' carbon pentose sugar forming the backbone, and a phosphate group connecting these units into a chain. So, why is this important? Without nucleic acids like DNA and RNA, life would cease to exist. They ensure that genetic instructions are preserved, transferred, and expressed across generations, enabling the continuity of life. Nucleic acids are more than just molecules. They are the foundation of life itself, dictating how organisms grow, develop, and sustain themselves. The experiment is also conducted so that we can study their genetic material and understand how their genes work. This process helps in areas like plant breeding, creating disease-resistant or drought-tolerant crops, and identifying plant species. It is also crucial for research like gene sequencing, genetic modification, and detecting pathogens to ensure crop health. Ultimately, DNA extraction allows us to explore the molecular blueprint of plants, advancing science, agriculture, and biotechnology. Let us now proceed to the experiment itself. But first, let's discuss the objective of the experiment. The group's objective is to successfully extract DNA fibers and explore the genetic information encoded in the plant's DNA. For the materials we will be needing for this experiment, we have the beaker, Bunsen burner, filter paper, funnel, graduated cylinder, iron ring and stand, mortar and pestle, pasture pipettes, serological pipette, steering rod, test tube rack, thermometer, water bath, wire gauze, and wooden stick. For the sample and reagents that we will be using, we have the banana where we will get the DNA, but you can also use an onion, dishwashing liquid which will break down the cell membrane, EDTA and sodium citrate as the lysis buffer, NaCl that will neutralize the charge of the DNA, papain as the protease, or you can also use bromine to degrade the protein from the banana sample. Lastly, we have 95% ethanol that is soaked in an ice bath, which will precipitate and lower the solubility of the DNA out.
Moving on to the procedure, let's first begin with Part 1, Preparation of Lysis Buffer. In a clean beaker, we successfully dissolve the following components in 50 ml of distilled water: 2.5 grams of SDS, 0.44 grams of sodium chloride, 1.94 grams of sodium citrate, and 0.5 grams of EDTA. The resulting solution was labeled as lysis buffer for use in the DNA extraction process. Next, the Part 2, DNA extraction procedure. Part 2, DNA extraction procedure. Step 1, preheating the lysis solution. We preheated 50 ml of the prepared lysis buffer to 60 degrees Celsius in a water bath. Step 2, mixing with plant sample. After reaching the desired temperature, we added 25 grams of plant sample to the lysis buffer and stirred the mixture continuously for 5 minutes. Step 3, adding papain. Following this, we incorporated 1.5 grams of papain into the mixture and continued stirring for an additional 10 minutes, ensuring that the temperature remained at 60 degrees Celsius throughout. Step 4, cooling the mixture. After mixing, we cooled the flask in an ice bath for 5 minutes, swirling occasionally to promote even cooling. Step 5, homogenization. The cooled mixture was then homogenized using a mortar and pestle until a uniform consistency was achieved. Step 6, filtration. We filtered the homogenized sample through 3 layers of filter paper, collecting the resultant filtrate in a test tube which was kept in the ice water bath. Step 7, DNA precipitation. Slowly, we added 15 to 20 ml of ice-cold ethanol down the side of the test tube, containing the filtrate. This caused the mixture to separate into two distinct layers. And lastly, step 8, observation and collection. After allowing the test tube to stand undisturbed for 3 to 5 minutes, we observed bubbles forming and DNA fibers emerging from the solution. Using a clean stick, we carefully spooled the DNA fibers. Data collection. In the plant DNA extraction experiment, we carefully recorded our observations at each step to see how well our method worked. After we added cold ethanol to the lysis buffer and banana extract, we noticed white, stringy clumps forming, which showed that DNA was present. However, these clumps were thinner and cloudier than we expected. Made sure the lysis buffer was heated to 60 degrees Celsius before adding the minced banana sample and stirred it for 5 minutes. After adding 1.5 grams of papain, we stirred for another 10 minutes. We then cooled the mixture in an ice bath for 5 minutes and homogenized it before filtering through 3 layers of filter paper into a test tube kept in an ice water bath. When we slowly added 15 to 20 ml of ice-cold ethanol, we saw 2 distinct layers form, confirming that DNA was precipitating out. After letting the test tube sit for 3 to 5 minutes, we observed bubbles and emergence of DNA fibers, although they were not as thick as we had expected. Overall, our data collection involved recording key changes throughout the experiment process. These observations provided valuable insights into the yield and quality of extracted DNA and informed our results and discussions. Proceeding with the results and discussion, this part we're going to discuss what the expected results are, and it is going to be followed by the actual results that we have observed. So the expected results for this experiment: in the DNA extraction experiment, we anticipated that the addition of cold ethanol to the banana extract would yield thick, visible strands of DNA that could be easily spooled onto a stick or pipette. We expected the DNA to appear as distinct, robust fibers, indicating a successful extraction process. This expectation was based on the previous studies and protocols that typically result in clear, well-defined DNA precipitates when using effective lysis buffers and proper precipitation techniques. Well, the actual results are contrary to our expectations, as upon adding the cold ethanol down the side of the detergent and banana extract solution, we observed the formation of white, stringy, and cloudy clumps. While these clumps did adhere to a wooden stick when swirled, they were not as thick or distinct as anticipated. Instead, they resembled a cloudy liquid with an evident layer that could still be considered DNA. This outcome indicates that DNA was successfully extracted. To end this presentation, our recommendation is to enhance the efficiency of lysis, and we can use other mechanical methods such as using blender for grinding. This approach makes it easier to break down cell wall of plants, making the process less time-consuming, although close observation is necessary to prevent overheating, which can harm DNA.
Now that we are done with the results and discussions, let's recap the key points we had earlier. Let's begin by exploring the basic role of nucleic acids, particularly in DNA in all living organisms. Next, we discussed the methodology of the experiment, which aimed to extract DNA from plant samples successfully. We also prepared a lysis buffer to break down plant cells and isolate DNA despite facing challenges such as the expected thickness and clarity of strands. Our observation indicated that we were able to extract successfully. According to our findings, we observed a thinner and cloudier precipitate rather than the thick, clear strands we thought it would be. This result implies that our extraction technique was successful, although it might require additional improvement for increased yield and clarity. In conclusion, the experiment not only deepened our understanding of the DNA extraction process, but also highlighted the importance of refining techniques to improve results. Extracting DNA from plants is vital for advancing research in genetics and agriculture, paving the way for innovations such as disease-resistant crops and improved plant breeding methods.